Unravel is going to present next. We have Dr. Richard Novak, and then we will do a joint Q&A for both of talks. So please welcome Dr. Richard Novak.
All right. Hi, everyone. I'm Richard Novak. Really wonderful to be here. I also want to introduce three of my colleagues who came with me. So we have Frederick Vigneault, co-founder and chief scientific officer, Eleni, program manager for rareSHIFT. I'll tell you a little bit about that program. And then Bridget. And so she is the one who handles a lot of the clinical samples, in addition to our tadpole models. And so you can ask about any of that. So go find them. But really, honored to be here. I love going to conferences that have families because ultimately, as you'll see, that's why we do what we do, and it's just also inspiring to listen to everything you've done and the challenges you've overcome. And our hope is we can make your lives a little bit easier. And so, what I'll talk about over the next few minutes is just discovering and validating treatments for OCNDS and how we go about that. So disclosures, hopefully it's pretty self-evident that I come from Unravel that has logos everywhere. So we're a biotech company. We spun out of Harvard about 4 years ago with really the idea that we need to change how we do drug discovery and drug development. And we've been focusing almost exclusively on rare disease. And so just to kind of get everybody on the same page, this is very, very generally how drug development works nowadays. So you start with a target, and this is thought what a target is what people think will actually help treat the disease. So if you can hit that target, it will treat the disease in some way, right? But of course, as you heard from Dr. Chow, you need some sort of intermediate steps to actually see, well, does it really work before you go into the patients, right? You want to not take that risk unless there's some evidence for it. So the mouse represents, preclinical models. It could be cells from patients, and you'll hear about that from Dr. Hunneman in a moment. Could be flies, could be mice, could be you name it. In our case, it'll be tadpoles, so little baby frogs. And then it goes to patients, right? Then you can finally ask the critical question, does it work? And then, of course, you have the approval process with the FDA or whatever the regulatory agency is. So we'll do a little activity here. And it's after lunch. We'll wake everybody up here. So raise your hands if you agree with what I'm saying. So this process works 100% of the time. Good. Nobody should be raising their hand. Good. 50% of the time. 25. 10, 5, 2, 1, 0. It never works. All right. So if you were below 10, you're right. General statistics are about 5% successful, but there's a caveat. If you're hitting a new target, it's only about 2% successful. And you guys are not lucky. In the neurospace, it's sometimes 0. So even the people who raise their hands for 0 were actually right. But we're here to change that, right? This is horrible. This doesn't work for patients. So what we started at Unravel is really working with patients directly. Solve for the problem you want to solve. So my background is actually more in engineering. If you want to build a bridge, not a car. And so we said, let's start working with patients directly. And it's patients, not diseases, which are these abstract concepts that actually respond to treatment. And so there's actually a picture from our office. A lot of wonderful families that we work with send us pictures of their kids. And really is just an inspiration for us. This is why we do what we do. It's why I get up in the morning. And this quote here really is from, I think, 1820. So this concept that you treat the patient, not the disease, it's been around for a while, and we're basically trying to come back to that. And so what we did to that is take that same order that you saw before, or same sets of activities, we shuffle them around, and we found that we start with patients because that's the problem we want to solve for, right? And this is why all of you guys are here, and this is why my team's here. So we can actually ask, what do you want us to do? What will actually be impactful? Not what we think, because we don't know anything. We can help you solve the problem, right? And so we developed a way where we can work using AI, using these advanced disease models to go backwards. Start with patients, and then go into animals, go back to patients, and then at some point we can figure out a target, we can find these mechanisms. But that's really the deep science. Ultimately, it's about function first. Does it actually solve a problem? And so we work with this concept of a patient, and a patient is not the gene that you're diagnosed with. That's very important. That matters, absolutely. That's a very critical piece. But there are, about 25,000 other genes in your body. They do stuff. Um, there's the environment. What you had for breakfast is impacting who you are right now, right? If you had coffee, you're probably a little bit more perked up. Um, other medications, other things like that, and then time. A patient at birth is different from a 10-year-old. And we saw that from Dr. Chung's presentation. There was this trajectory of increased skills and other aspects. And so to understand that, we collect information directly from patients, right? This is the problem we're trying to solve is whatever that patient's state is. We're trying to make that more or less affected. How do we understand that? And to us, the answer lies in RNA. So you've probably heard about DNA, which is what your cells have, the instructions for making RNA and proteins, and that's how these patients are almost always diagnosed. It doesn't change, except in cancers, but it really doesn't change. RNA changes when you wake up versus when you go to bed. Your RNA is quite different. If you eat something, your RNA changes in certain tissues. And so we can start using that as a way to understand these very complex things that are patients. And so we built a model of human health using machine learning and some other approaches. And now we can start working with a real patient computationally and test all these different drugs and just see what happens, right? So we don't know what we don't know. Let's try it. And really, the whole idea is this concept of personalized medicine. It's going back to the individual patient. I don't really care if a drug works in a whole population. It almost never does. But we can work with individual patients. This is what clinicians do. This is what each of you do with your own kids. And so what we have is the patient RNA signatures, very complex, data that we get from each patient and a matched control. And we have similar data from drugs, molecules, about 40,000 molecules in our library. And these are all purchasable. Many of those you could actually give to a patient today. And we run it through our platform, and I'll talk a little bit more about what that looks like. And then the outputs are, these couple different options. Personalized therapies. So a repurposed drug, like you heard Dr. Chow's presentation, very similar concept, but computational at this stage. We can also ask the question, can we treat the whole population? The answer may be yes. The answer may be no, but we could treat half of the population with this drug and half with a different drug, right? And so you hear all the time about patients. You might talk to someone who has a very similar kid, symptomatically, even sometimes the exact same mutations, and they may not respond to the same drug or same intervention, same physical therapy, right? Why? Well, it goes back to that. It's not just the gene that's mutated, it's everything else working together. And then for us as a biotech, we care about new drug targets, and then also making drugs that are safer, right? So this is when we want to make new drugs. This is what really comes to mind. So what we built is, think of Google Maps, right? I'm sure navigated from A to B at some point using Google Maps or some similar software, and that's really what we have here for health and these disease states. Now we don't understand the disease states, but we do understand the health piece quite well, given all the data that we have, and so we can map it. We can basically say, okay, relative to a control, like let's say a same-sex first-degree relative is typically what we go with. How different is the patient, and very importantly, how do we fix it? Whatever the it is, right? It's a very complex set of challenges, but all I care about is going from A to B. Just like how you got here, into the hotel, you probably used Google Maps, or your Uber driver used Google Maps. And what we do, we just simulated all these different drugs, and we just asked, well, what happens? And we can rank them based on, it really got us to where we want to go, to it took us in the exact opposite direction. And it's very similar to what you saw in Dr. Chow's talk. Some drugs help rescue a symptom in the flies. Some drugs make it worse. We have essentially a computational version. And then we can find how we get there, those targets, and we can make drugs even better. That's more the biotech arm of our company. That's not really part of my talk here, but that's the end goal. And so really what we have here is a personalized medicine for all approach, because what we do to what we call digitizing the patients, how do we get that patient signature, is a nasal swab. And actually, some of you in this room may have actually participated in our study that I'll talk about in terms of the results. It's a nasal swab, just like COVID tests, except it has a special solution that really does a good job preserving the RNA. RNA is very unstable, but we work with a company that does a good job preserving it, and we can ship these sample kits all around the world. And we can basically use that to understand what's going on in this patient and their control, right? So they're about 50% similar in terms of their genome. And then we can simulate them using BioNAV, that's our Google Maps, and to ask, what's going on in the patient and how can we fix it, all right? And different patients may have different results, and that's okay. And so we've been working with patients literally all around the world. So thousands of samples. This represents hundreds and hundreds of patients. 30 countries, 6 continents, no penguins. And it represents about 70 disorders. So 70 different genes, heavily working with foundations that sometimes represent multiple disorders or multiple genes. And what's really exciting is that, just like in Dr. Chow's talk, the path to actually testing back in patients, so usually going through some intermediate model or some other preclinical test, but it's really fast. This can be on the order of a few months, not 5 to 10 years that you saw in the first slide. And we've had a number of clinical benefits, ranging from being able to walk, better communication, better behavior. Really, there's a number of effects that we've been able to observe. And I also want to point out this is vastly working with children. And I think with Dr. Chung's talk, you saw something similar. And so how can we test these drugs? Right? These are just AI predicted. I always say the predictions are only as good as the bits they're written on. We need to test in some sort of model system. And so for that, we developed something called squishyware. It's not it's not software. It's not hardware. It's squishy. And so we use gene editing to basically put that same sort of mutation that your child might have into tadpoles when they're 4 cells in size, and they grow up to essentially being a model of a patient, like a patient avatar. And they're very, very different. This comes from different, it's 4 different females are used in each study. They're all different genetically. So it represents kind of the whole population, the diversity that we have in this room is represented each dish that we use. And what we see all kinds of symptoms. And we can use computer vision and AI and other ways to look at, okay, what are these tadpoles doing that's maybe different from a kind of a controlled tadpole? And we can look at all kinds of things from movement, shape of these tadpoles to even sleep. They sleep at night like we do. And then importantly, we can stick drugs on there and see, does it actually fix any aspect, right? Maybe none, maybe multiple, but we have now a way of reading out what's going on. And so I can't stress this enough. This is a very different way of doing drug discovery. It's function first. To put it in very sort of stark terms, we don't care about how it works. We care that it works. That's the key. And we can always find everything else out later. And it is important to do the basic science, but we're basically flipping the narrative. And then what we're always focused on, we have a chief medical officer who couldn't be here today, but we're focused on what does the clinical study look like? And we work heavily with clinical teams, primary care to expert neurologists, to figure out how do we test this in the clinic. And it may be something very benign that could be readily prescribed, to heavy-duty cancer drugs that maybe at low doses could be quite effective, right? And that needs a full clinical trial, a lot more data, and so it's very case by case. And so what have we done for CSNK2A1? All right, so this is really work driven by many of you, and Jen Gabrielle, very graciously funded this, and the foundation funded this, and the whole idea is to develop treatments for OCNDS. So the purpose was, one, let's generate data. We really don't understand what's going on with patients. Let's get that molecular information as soon as we can and start working with it. Let's predict these drugs, right? What could rescue some of these effects? And then let's actually further drive them, go into screens and see what happens. So huge thank you to the 22 participants in the study. It was 10 patients with the K198R mutation, and the foundation felt that was a really nice way to start understanding, especially the subtle differences between patients with identical mutations, right? So this is all the other stuff. It's not the diagnosis. It's all the genes, environments, and time for each patient, and what's similar and what's not. And then 47G patient, really to complement that. So huge thank you. I know it's a disruption to your lives to have this extra step involved, but just rest assured it is extremely valuable. So thank you. So what did we see? So this is a plot just really focused on the colors. It's correlation scores, but basically it says how similar are the patients to each other, not in terms of what caused the disease, but in terms of how would you treat it across hundreds or thousands of drugs. And so the more red it is, the more similar the patients are. The more blue it is, the more opposite the patients are, right? So what would be good for one patient would actually be worse for the other. And all but one of these people have the exact same diagnosis, right? And yet there are these two groups that you start seeing. And it's very, very sensitive to we see a lot of what goes on, medications that people are on. There, you might notice that top line, it says once UB162 alt control, and there's also UB162 in the middle. We realized this was sort of an outlier in many ways, and so we started looking at what's going on. Well, it turns out the control had undergone a medical procedure, and they were on these medications, and turns out that completely threw the results for that patient. So we adjusted. We can work with that, but all of a sudden that patient belongs in that main group. But what this tells us is that even though these are patients with identical diagnoses, they're not identical in terms of how you would treat it. And when you talk to business people in biotech, they're like, well, that's terrible. You're now making a market even smaller. My point is, you can make drugs that work better, right? And that's what we do. But what's exciting, we've been actually able to, again, thanks to the foundation, work with many of these other researchers that you've heard about or will hear about, and we can start seeing from the tissues from these other model systems, how similar are they to the patients that we've worked with? And so there's several mouse brain samples in there, and some patient lines, cell lines from TGen, and now we can start seeing what's going on. And again, you see these two general groups emerge. That tells us something. There's some overall patterns and differences that we should be aware of. But we can do even better, right? So the challenges in rare disease are there's relatively few patients. The people who care about the money side, they see lack of dollars, they're not interested. That's unfortunately the kind of brutal truth. But we decided to change that. The whole idea is, well, why are you limited to a very rare disease? If you go to a clinician, they might give an OCNDS patient the same anti-seizure medication they're going to give to a Dravet patient, potentially, right? Why not think of that as one larger group of patients based on drug response, not based on the diagnosis? And all of a sudden, this opens up possibilities where we can syndicate drug development. And so now each foundation, each family doesn't have to worry about a drug for their little population. We can still have the same effective drug, but now it covers multiple groups. And so this actually results that we did, thanks to Jen Gabrielle's prompting, looking at similarities with SYNGAP. So a very different disorder, but there's some rationale for why it scientifically makes sense. And so we looked at it. Well, it turns out you again have maybe three groups. There are actually three groups in the SYNGAP population, but the OCNDS ones map perfectly right on there. So what it tells us that we still have those kind of two or so groups, but it's the same two groups in OCNDS, same two groups in this other disorder. So now we've dramatically increased the number of patients. And that can accelerate clinical trials, accelerate funding, accelerate interest and attention, right? And so all of a sudden, there's just much more work that could be done faster. And that's what we did on the computational side. So now kind of going back to how do we test it, the proof's in the pudding, or tadpoles. We made a tadpole model, actually two tadpole models. One is the KD, CSNK21 KD. Basically, we just reduced the amount of that gene being made. The other one is we reduce the amount of the gene being made, and we add basically the RNA that causes that K198R mutation. So now it's really that same population of patients that we saw earlier. And what we see here is we can look at how they swim. The tadpoles like to swim around a dish in circles, and they do that less. They swim across the dish. They don't move as much. There are all these issues. They also tend to be more on their side. So kind of their coordination and vestibular function is a little bit messed up. We can also look at their brains. So on the left, you see a normal tadpole brain. You can just look at it, and has different sections. Looks very different from a human brain, but it's there. And then we can look at the brain relative to how severely affected the tadpoles are in terms of their behavior. And we start seeing some of these misshapen brains, smaller brains, and it correlates with how severe the disease is. Even better, we can look at how's their sleep. Now, I don't know if the sleep in a tadpole is the same as sleep in a human, but we're trying to make, again, unhealthy tadpoles healthy again. And turns out at night, they don't sleep. And so we've heard that from the families here. So we have that in tadpoles. And then this is data that I got from the team flying over here. So this is very, very fresh. We looked at, again, the RNA expression profile of these tadpoles, and we wanted to see how different is the tadpole model relative to the controls. And then if we add one of the drugs that we predicted for the patients you saw earlier, Again, we worked with the foundation to narrow it down, and it's a prescription drug that you could get prescribed by a clinician. Does it help? And all we're looking for is essentially that blue area on the bottom. That's the disease state. Does it move up? And the we see is the orange or the orange dots are the treated tadpoles with this drug, and then now they're actually much more similar to the controls. So that was really exciting to see. This is very much work in progress. This is not final by any means, but we're actively working on this. But just to summarize, we generated RNA-seq data for researchers to use from you are part of these studies, and the controls. I always have to say the controls are just as much part of the study as the patients. We predicted a bunch of drugs that could be clinically translated, and we have 2 tadpole models that are avatars of the patients in this room. And so with that, I want to thank you for being here and for sharing your stories. It's extremely moving. And this is what keeps us awake at night in a good way to try to solve these problems. So thank you so much.